All right, guys, in today's lecture, we're going to be discussing the details of DNA replication. So if we're replicating DNA, where are we? So remember, there's all that stuff where cells are dividing and doing all of that stuff, whether it is mitosis or meiosis. But if we are replicating DNA, we are an interphase, right? And if we're an interphase, what part of interphase do we actually replicate DNA? It's not G1. It's not G2. Today's discussion is happening basically entirely within S phase of interphase. So there are a few, let me go back actually. There are 4 principal enzymes about DNA replication specifically that I want you to know. Okay, and I'm going to discuss a 5th one, but it's sort of at the very, very end of DNA replication. But the 4 that you need to know are helicase, which unzips the DNA, breaks those hydrogen bonds; polymerase, which actually builds a new DNA strand; primase, which helps let polymerase know where to go; and ligase, which stitches together back what are called the Okazaki fragments, which we are going to discuss here shortly. Okay, so those are the 4 main ones that I want you to know. I don't want you to think that that's all there is in DNA replication, but those are the main ones that we're going to be discussing. Okay, there are actually many more. So remember, enzymes lower the energy required for a reaction to take place. So if you didn't have these enzymes, DNA replication really couldn't happen. The other thing that I'd like you to remember is the basic structure of DNA. We've already talked about this way back in the first section, but you should know that there's sort of this deoxyribose sugar, and then a phosphate group, and then another deoxyribose sugar, and then another phosphate group. And these deoxyribose sugars are attached to these nitrogen or nitrogenous bases. And those nitrogenous bases pair to another strand of DNA that's heading the opposite direction. It's what's referred to as anti-parallel. Okay, so basically so far what you need to know, the basic structure, okay, deoxyribose sugar, phosphate group, deoxyribose sugar, those are attached to base pairs. Those base pairs are attached to one another through hydrogen bonds. Okay, and as we're going to see in just a minute, the helicase is going to come through and break those hydrogen bonds up. The other thing, so I put deoxyribose and ribose, but really what you need to be focusing on is this deoxyribose, because DNA has directionality to it. Okay, so I'm not going to ask you about these specific chemical details, but you need to understand that DNA has a direction to it. Okay, and the way that this directionality is determined is by numbering the carbons on the DNA molecule itself, which might seem kind of random, but this is based on chemical conventions. So if you don't quite get this, you can blame the chemists. But basically what I want you to know is that DNA has directionality. I don't necessarily need you to be able to number carbons or anything like that, but if I say something like 5' to 3', I'm just talking about a direction. Okay, I might as well be saying left or right or up or down. Okay, so DNA has a directionality, and I'm going to be referring to it using the carbons that are actually on the sugar. You don't necessarily need to know that. I'm not going to test you and be like, what's attached to the 3' carbon or anything wild like that. Here's a little segment of DNA. Again, this should all look somewhat familiar to you by now. You've got this is representing a phosphate here with the circle. This is your deoxyribose sugar. Here are your bases. So the first enzyme involved here is helicase. Helicase is going to come in and it's going to break these hydrogen bonds. The reason that the directionality was important, and what I want you to notice here, is that there's a 5' end on the left of this strand and a 3' end on the right of this strand. Notice that up here it's flipped. And so when I'm talking about what I'm referring to, okay, so you have one segment of DNA going this way, you have one segment of DNA going this way. And they're going to line up with one another, and those complementary base pairs are going to get together. So onto helicase. Because remember, what we're trying to do is we're trying to make a copy. And if these things are all too closely associated with one another, the other enzymes can't get access to them. So helicase, you can think of it like the icebreaker. Think about if you've ever, back when we were able to go to parties, if you've ever encountered a group of people all standing in a really, really tight circle and you need to talk to one of them, okay, helicase is just going to be the shoulder in, okay? It's going to break those hydrogen bonds so that we can get access to these base pairs so that we can make copies of them, okay? The next enzyme that's going to be involved here is DNA polymerase. Once you've exposed those strands, DNA polymerase is going to be able to come in and actually add base pairs. Okay, so that we're winding up with a copy up here and a copy down here. Now the thing about DNA polymerase, it's a really great enzyme, but it's not smart. It is just an enzyme. Okay, so the three things to know about DNA polymerase is it adds DNA base pairs. That's its job, is to build a new strand of DNA. That's like the main thing. The other thing to know is that DNA polymerase only can work in one direction. So I was talking about 5', 3'. I don't necessarily need you to know which direction it can work in, but you need to know that DNA polymerase only goes one way. Okay? It can only work one direction. The other thing you should know is that DNA polymerase also doesn't know where to start, which is where our next enzyme is going to come in. Okay, so what do we know so far? We know that DNA helicase breaks those hydrogen bonds between base pairs, unzips the DNA strand. We know that DNA polymerase is going to come in after that strand has been opened up, and it's going to add new base pairs. We also know that DNA polymerase only can work one direction, and DNA polymerase doesn't know where to start. In the next lecture, we're going to be finishing up this process. Hopefully we can get it done in one more lecture, and we're going to talk about the other enzymes involved and how polymerase contends with this problem of not knowing where to start and only being able to work in one direction.